Okay, good afternoon, everyone. Thank you, ma'am, for a nice introduction. And so today's session is about functional connectivity changes in Alzheimer's disease, right? Everyone has looked at uh, uh, the schema. So what I thought I'll do is I'll provide you with the overview of Alzheimer's disease. That will hopefully set a stage for Professor Viswal's talk about functional connectivity changes in Alzheimer's disease. So, before talking about Alzheimer's disease, I thought I'll briefly mention dementia because dementia is an overarching term which covers a lot of diseases, including the Alzheimer's disease. So, by definition, dementia is a chronic condition um, with impairment in memory, language, problem solving, thinking, and other abilities that are severe enough to interfere with daily activities. So, you all have noticed that we all have problems with memory at times, forget somebody's name, some friend's name, or forget something or the other. Where did he put the keys in the morning, or this and that? But unless it really interferes with your daily activities, it will not be characterized as a clinical condition, it will not be characterized as dementia, right? So that's the operational trait in the sense that it has to interfere with the daily activities. Now worldwide about more than 7 million cases of dementia have been identified. This is a little older, and about 4 seconds it takes for new dementia cases somewhere in the world to appear. So you can imagine, by the time we finish this talk, there will be several people who would have had developed into condition would have developed into dementia uh, somewhere in the world. So yes, 2021 we had about 55 million people with dementia, which is projected to increase to 2050 by 25 to about 135 million, which is a pretty big number considering the burden that it puts on the individual, on the caregivers, as well as on the society and the nation. So this is a nice graph showing how cases of dementia are projected to increase over time. This is about high impact, high income countries and low and middle income countries. You notice that in 2015, yes, there were a little more number of people with dementia in low and middle income countries than the higher income countries. But over time, it's projected that the increase that's going to happen, the estimate is that the increase that's going to happen in dementia cases is going to happen more in the low and middle income countries than in the high income countries, right? So you can imagine that it will put additional burden, right? And low and middle income countries are less equipped to deal with this kind of conditions. And yeah, sixth leading cause of death in the US. Yes, so dementia, there have been a lot of studies about dementia as well as Alzheimer's disease in India as well. And you will notice that the prevalence of dementia varies a lot between different cities or different regions of the country, right? For example, in Cochin, Kolkata, it's about what 1.5%, but in Trivandrum and Cochin and Pune, it will be about 4.5%, right? So that's actually translating into the entire population. That turns out to be pretty big difference, right? It's not clear why that difference is there, right? But one possibility is that anyway, if you consider with the US, which is more than 65 years of age, if you consider that will turn out to be about 8%. So none of the studies in India have shown that number yet. It's relatively lower percentage of dementia cases in India, but still varies a lot between different regions. It's unlikely that people in Kolkata have certain traits or certain advantages such that they develop less dementia. It seems more that different investigators have used different kinds of criteria to define dementia, and that's why the percentage is so different between different regions. right? So yes, there was the dementia science program started to normalize all these things in India also. Yeah. So yes, dementia, majority of the cases will be Alzheimer's disease, about 70% of the cases will be Alzheimer's. 20% of the cases will be vascular dementia, so that's the second leading cause. Frontotemporal dementia about 5%, and DLB, that is dementia with Lewy body, about 5%, right? Some people characterize 1 or 2% here and there with the mixed dementia when it's not possible to classify them into any of these groups, but that doesn't matter. The point is, majority of the dementia cases, about 70% of them are Alzheimer's kind, right? So this is how the study on Alzheimer's started actually. In 1901, Alois Alzheimer noticed a patient, Auguste Ditter. She was in an asylum in Frankfurt, right? And she showed severe symptoms, including the memory loss. Basically, what we understand now as advanced stage of Alzheimer's disease is what she was suffering with at that time, right? So she passed away a few years later. In 5 years, she passed away in 1906, and then Alois Alzheimer conducted the post-mortem study on her brain and figured out that there are plaques and neurofibrillary tangles in the brain, and that came out to be the defining feature of Alzheimer's disease, right? So nowadays, if these are the fundamental criteria to identify Alzheimer's disease from other diseases, but you can imagine that these studies cannot be done until the person has passed away, right? Post-mortem studies. There are ways to do imaging studies with the plaques in the human in the live human also, but uh, the confirmation of Alzheimer's disease this is how the scientific community believes, not the clinical community, that confirmation of Alzheimer's disease can be done only after post-mortem studies where you see the plaques as well as the tangles, but the plaques can be imaged, tangles not. So as far as incidence of Alzheimer's disease is concerned, incidence is defined as number of new cases, right? So let's say today certain number of people have some problem, and after a year how many new people have that kind of problem will be defined as the incidence, right? But taking a cross sectional overview, how many people of the class of students have very high marks, that will be kind of rough example, that will be the prevalence of it, right? So prevalence refers to the number of people in the population living with the disease at a particular point of time. Now point of time is not going to be that precise, it takes some time to do the study. So over a period of few months to a year, whatever possible. So in US it was about 3 million people, and it's supposed to be 13.8 million people. As far as the incidence is concerned, about 469,000 people over the age of 65 years in US had dementia, right, per year. It's classified into two groups. One is referred to as the FAD or familial Alzheimer's disease, and the second one is referred to as the SAD or sporadic Alzheimer's disease. The first one is also referred as early onset Alzheimer's disease, and second one as late onset Alzheimer's disease, because the age of onset under these two conditions differs, right? But majority of the cases are SAD kind, sporadic kind, and sporadic simply says that okay, yes, I don't know what the cause is, right? But in familial cases which are less than 5%, these are due to mutations in three genes, particularly amyloid precursor protein, presenilin 1, presenilin 2. I hope that everybody understands what mutation means. Otherwise, it simply is that you have a particular protein sequence, right, amino acid sequence. If the amino acid sequence is changed and it creates a different kinds of function for the protein, then that is the mutant protein, right. So it will not be able to do the same function that it was able to do earlier, right. So, its early onset before the age of 65 years, 30 to 60 years is common, and majority of the cases are a load kind, the late onset kind, which occurs little later in the life, that is more than 60 years, and yes, precise cause is not known in these cases, right, but there are several risk factors that have been identified. So, before someone develops full fledged Alzheimer's dementia, people have this condition that is called mild cognitive impairment, MCI, right. Now, everybody who has MCI does not convert to dementia, does not convert to Alzheimer's disease in the later stage, right. So, some percentage of people convert. For example, this just says that over a course of a year, about 24% will convert to Alzheimer's disease, and about 48% over the course of 2 years or so. So, there will be a lot of people who will never convert to Alzheimer's disease, right. So, this is what is considered kind of window of opportunity to do something such that the conversion of MCI to full fledged dementia or Alzheimer's disease can be prevented, right. So, a lot of studies are trying to focus on this MCI period, trying to find out what kind of biomarkers might be that can predict whether a person is going to convert to Alzheimer's disease or not, right, and obviously that will be a very helpful finding. If we talk about the prevalence age wise, by about the 65 years of age, about 1% of the people have Alzheimer's disease, but by 95 years of age, about 50% of the people will have Alzheimer's disease, right. So, you can imagine that, the uh, lifespan of our lifespan has increased considerably over the last few decades. So, you can imagine that more and more number of people are going to live to the advanced stage, and that is a good thing, but at the same time it is going to create a different kind of problem, that is we are going to have a lot of neurological diseases including Alzheimer's disease, right. So, yes, lot of different kinds of policies are needed to tackle this issue. Now, going to the pathology of Alzheimer's disease, as I mentioned earlier, plaques and neurofibrillary tangles. Plaques are made up of a particular protein called amyloid protein primarily. So, it is called amyloid plaques, and it is extracellular, meaning that it is present outside the cell, right. The second defining feature is presence of these neurofibrillary tangles, which are present inside the cells. So, they are intracellular, right, and they are basically hyperphosphorylated form of a particular protein called tau that is present in the neurons, right. So, yes, a lot of changes have happened in tau, and it forms kind of aggregates, and that becomes the neurofibrillary tangles, right. This is the schematic showing amyloid plaques in between cells and neurofibrillary tangles inside. This is the actual staining actually showing that yes, amyloid plaques are present outside the neurons, but this is the cell body boundary of a neuron, right. It is a kind of pyramidal neuron, so pyramidal shaped cell body. The tangles are present inside in this, right. And in severe cases, people have noticed that there is lot of neuronal loss, right. So, neurons die, right, and neurons die, that means the size of the brain is going to shrink a lot, right, because now there are less number of cells and there is increase in the ventricle. So, that is the kind of brain that we can see as compared to the healthy brain in severe Alzheimer's disease cases, right. So, that is the pathology. Now, we think about what causes Alzheimer's disease. So, actually what some 125, 130, 125 years or so it has been since the disease was identified. We still with confirmation cannot say this is what causes Alzheimer's disease, right. There is good idea about it, but still some people do not agree with that. But primarily there are two hypotheses. One is the cholinergic hypothesis and the second one is amyloid beta hypothesis that kind of defines how Alzheimer's disease can be caused, right. cholinergic hypothesis simply says that acetylcholine, neurotransmitter acetylcholine, is not present in the enough quantity in the brain, right. So, obviously, it is a neurotransmitter, so it has to be synthesized, it has to be released, right. So, less amount of neurotransmitters you can have because of a variety of conditions. That is, there is problem with the synthesis of the enzymes that make the acetylcholine, or the release of the acetylcholine is restricted, something. But the end result is that there is less of acetylcholine in the brain than normally should be, right, and that causes the Alzheimer's disease. And there is I must say pretty good evidence for this as well, right, clinically. But most of the people believe, including me, that Alzheimer's disease is caused by the particular protein called amyloid beta, right. And from various studies, this is the kind of cascade of events that one has come out with, saying that these are the events that happen during Alzheimer's disease development caused by amyloid beta, right. So, mutations in presenilin 1,2 APP that we talked about earlier, that will lead to early onset Alzheimer's disease. ApoE4 is a particular protein called apolipoprotein. Don't worry too much about the name, right. It's just a protein that carries cholesterol in our body, and it exists in a few forms. If the particular form ApoE4 is there in our body, then we are susceptible to developing Alzheimer's disease, right. Trisomy, I think all of you are familiar with the Down syndrome condition, right. It's 3 chromosomes of chromosome 21 are there. So, trisomy 21 it's called, and it happens to be the it's just the situation that the particular gene that codes for amyloid beta, the precursor protein, amyloid precursor protein, is present on chromosome 21. So, you can imagine if there are 3 copies, then more of APP will be produced, right. It will be more than the 2 copies. It's simple, 1.5 times amyloid beta will be produced, and that will lead to aggregation of the amyloid beta in 2 forms: soluble oligomers and plaques. Plaques are kind of the structures that don't dissolve, right. They are kind of solidified structures. Oligomers are small molecules that can be dissolved in different kinds of solutions. So, they cause synaptic dysfunction, and synapse is the, I am sure you know, but I just emphasize again that synapse is a point of communication between neurons. So, it is a very important in the brain. So, synaptic dysfunction happens, neuroinflammation happens, and that leads to the formation of neurofibrillary tangles, right. So, according to this hypothesis, actually combined with other cases, there are certain conditions in which neurofibrillary tangles can be formed on their own, but otherwise most of the time it is thought that they are formed because of amyloid beta overproduction, right, and that will be lead to neuronal dysfunction, neuronal death, and eventually that will result in dementia, right. So, this is the amyloid cascade hypothesis that has received more support, although nobody discounts completely the cholinergic hypothesis, right. Okay, so, this is how amyloid beta is produced. Remember, the plaques are made of amyloid beta. So, amyloid beta has to be overproduced in the brain, and this is how it is. There is this protein called APP, or amyloid precursor protein, right. So, this part of the protein is the one that makes the amyloid beta, right. So, if the protein is this long, this part will be the amyloid beta, right. So, it can be cleaved by an enzyme called alpha secretase, which cuts it here, and then gamma secretase cuts it here. So, you realize that now amyloid beta is in 2 parts, 2 separate parts, and this does not lead to plaque formation because they can't aggregate, right. But in Alzheimer's disease cases, the APP is cut by beta secretase here and gamma secretase here. So, entire amyloid beta is produced, and that's a very sticky protein. So, that's why it forms the plaques or oligomers first and then the plaques, right. So, this is how the amyloid beta is produced In normal case, it will be this pathway that is followed, but in disease condition, it will be this pathway that is predominant, and one ends up with a lot of beta amyloid production. That, being a sticky protein, makes oligomers and then makes the plaques. So, that is how. So, yes, amyloid plaques and neurofibrillary tangles are the defining features of Alzheimer's disease, right. But there have been several post-mortem studies where people have noticed that some people have a lot of plaques, right, but actually they didn't have dementia when they were alive. So, we are talking about post-mortem studies, right. Or there are people who have very few plaques, but they have severe dementia. In other words, there is lack of direct correlation between the amount of plaques and the severity of the disease, right. So, that is when people started thinking, how is that possible? If plaques are the cause for the Alzheimer's disease, then having more plaque would mean severe Alzheimer's disease, but that does not sound to be true in at least some cases. So, that is when this idea of oligomeric amyloid beta came about. And oligomer simply means you have one molecule, that is a monomer, you put quite a few of them, 10, 15, 20 of them, that becomes oligomer, right. Most of the time this will be soluble in some kind of solution, solvent, but if becomes too big, then it makes fibrils and then it is not soluble, right. So, people thought that oligomeric amyloid beta, there were good indications to believe that oligomeric amyloid beta may be involved in early stages of the Alzheimer's disease, right. So, people tried to detect oligomeric amyloid beta in Alzheimer's patient's brain. Yes, they found it. They found it in the animals that are used as model system for Alzheimer's disease, and they can kill the neurons. They can inhibit a form of synaptic plasticity, that is long term potentiation, that is the synaptic mechanism of memory formation, and they by themselves can cause memory impairment also, right. So, this is just I think in one or two slides I will show that nowadays it is believed, actually it has been demonstrated, that oligomeric amyloid beta can do everything, almost everything that has been thought to be associated with Alzheimer's disease. For example, change in signaling molecules, right. So, when we some hormones are there, they do particular reaction in the body. So, that is the signaling event kind of. So, these can impair the signaling events. They reduce the synapse number in the brain, right, and that can lead to dementia, of course, in this case, and they reduce the dendritic arborization. So, that is less number of branches of dendrites will be formed. Dendrites are the receiving end of the neurons, right. So, I think you are familiar with dendrite, soma and axon terminal. This is a kind of synaptic plasticity that is called long term potentiation, and higher long term potentiation means good memory formation, that is extrapolated to, and lower long term potentiation LTP will mean no memory formation, right. So, you will notice here that, so this is done with the hippocampal slices, because hippocampus is involved in different kinds of memories. So, if you want to do this kind of study, you have to take out the hippocampus and then cut very thin sections and then do this electrophysiology kind of studies, right. But the message here is that the amyloid beta oligomers inhibit long term potentiation, and by extrapolation they will inhibit memory formation, right, and that is the property in Alzheimer's. Actually, and they kill the cells, they kill the neurons. But the conclusive experiment with respect to amyloid beta and memory formation has come from the experiments that have just injected the oligomeric amyloid beta in the cerebral space, right. So, ICV injection right, intracerebroventricular injection. So, if you do that, then the animals show long term memory impairment with oligomers and not with others. So, that kind of conclusively established that amyloid beta oligomers are very important species that cause Alzheimer's disease, right. Plaques in the Alzheimer's disease cases can be imaged, right, and the primary imaging used to be, even now it is, that the Pittsburgh compound, which is an radioactive analog of a thioflavin T, right. So, thioflavin T, don't worry about the name and all that, it is a compound that binds to the amyloid plaques, right. Now, if you can make it radioactive, that means it will emit radiation, some form of energy that you can detect in some ways, right. So, that is the advantage here in using the Pittsburgh compound V, which is radioactive analog of this. It is used in the PET imaging, positron emission tomography, and here is the Alzheimer's disease case. Here is the control brain. You will see that control brain has very little signal, if at all, right. That is the binding of thioflavin T to amyloid plaques is extremely little, if at all, in the normal condition, which essentially says that there are no plaques to which the thioflavin T can bind, right. But in Alzheimer's disease, different brain regions show that some have very high binding, means there are a lot of plaques in those regions, and some have less binding. The yellow ones show the less binding, they have less number of plaques there, right. So, this is how imaging is done in the Alzheimer's disease cases using Pittsburgh compound B, right, and the PET imaging. Okay. Biomarkers, everybody familiar with, right. Biomarkers are the kind of markers that will, with very high confidence, predict that the person is suffering from this particular disease, right. For example, if somebody has very high glucose level, one would, constant glucose level, one would say that the person is diabetic, right, hypertension, high blood pressure and things like that, right. For most of them we can find. In case of COVID, you are all familiar with that the RT-PCR was done to detect a particular kind of antigen, and if that was present there, then one would say that yes, the person is suffering from the virus, in viral infection, right. So, that was the biomarker. Unfortunately, this kind of biomarkers are not available for Alzheimer's disease, although some of them have been tried, right. People have found that in the CSF, and the CSF is, what is CSF? Cerebrospinal fluid that circulates in the brain, right. So, it is kind of, for a lot of things we do, a lot of conditions we do blood analysis, right. For a lot of conditions, the brain conditions, CSF analysis is kind of equivalent, although not exactly the same, right. So, one can take out CSF and measure different, molecules in the CSF, and people figured out that the controls which do not have Alzheimer's disease have higher level of amyloid beta, and the people who have Alzheimer's disease have lower level of amyloid beta, right. Is this clear? Normal people will have higher level of amyloid beta, but Alzheimer's disease patients will have lower level of amyloid beta in the CSF. Now, we earlier talked about that amyloid beta is the one that causes Alzheimer's disease. So, it is kind of confusing, right. The reason is that in the Alzheimer's disease cases, although you will have more of amyloid beta, that is in the form of plaques and not free in the free form in the CSF, right. So, that is why the, although it will have more amount of amyloid beta as such, but it will be in the form of plaques which cannot be detected in the CSF. So, that is why the level goes down. If there is neurofibrillary tangle in the Alzheimer's case, right, then tangles are, the tau protein is present inside the cell, we talked about earlier also, right, neurofibrillary tangles are present inside. But if cell death happens, then these proteins will come out from inside the cell and they can be detected in the CSF, right. So that is why the CSF shows higher level of protein of tau than the amount of tau in the control conditions. But you will notice, and yes, oligomeric amyloid beta also people have looked at higher level in the Alzheimer's disease cases and lower level in the control populations. But yes, higher level, lower level statistically significant difference is there, but actually if you look at the distribution, it's pretty much overlapping in a lot of cases. So that's the reason, although these are good indicators, they cannot be used as biomarkers for Alzheimer's disease, right? And yes, currently there is no biomarker that one can use for Alzheimer's disease. There are several risk factors for Alzheimer's disease. Age is the biggest risk factors. Anybody more than 65 years of age is at risk of developing Alzheimer's disease, right? Uh, if the person has mutations in the 3 genes that we talked about, then it will be earlier. The person will develop Alzheimer's disease earlier. ApoE4 allele. So again, ApoE4 is a particular protein that transports the cholesterol, right, for liver metabolism, and it exists in 2-3 forms. One is called ApoE epsilon 2, epsilon 3, and epsilon 4, right. If some majority of the cases, 75 to 80% of the people in the population have epsilon 3 allele, which is kind of neutral, does not confer any protection from Alzheimer's disease, does not confer any risk for Alzheimer's disease, right. But if epsilon 4 allele that confers risk, that is, if somebody has epsilon 4 allele, then there are chances that the higher chances that the person will develop Alzheimer's disease. If there is only one copy, remember we have 2 chromosomes, pair of all the chromosomes. So one copy is present of some gene in one chromosome, and second copy is present on another chromosome, right. So if somebody has one copy of epsilon 4, then has a 6% higher chance, 2 copies 7 to 22% higher chance of developing Alzheimer's disease, right. This epsilon 2 actually turns out to be protective, right. It will prevent the development of Alzheimer's disease, but the percentage of people that have is very low, about 10% or less than 10% people have epsilon 2 allele. So it does not really matter too much. Lifestyle risk factors, hypertension, diabetes, obesity, all these have been linked to development of Alzheimer's disease in the later stages, right. And there are good reasons to correlate the hypertension, the diabetes and obesity with the development of Alzheimer's disease in the later stages, right. There is good amount of literature about it. Okay. Finally, okay, coming to the treatment, there is no treatment, right. So there is no drug that you can take and you will be free from, I mean, you means anybody who is suffering will be free from Alzheimer's disease, right. That is the unfortunate part of it. And it is true actually for a lot of neurological diseases. For Parkinson's, we have good one medicine, but L-DOPA, but that also in the later stages is not so effective. So that's a problem for most of the diseases. But in 1966, donepezil was approved, which is acetylcholine esterase inhibitor, right. It was approved for mild to moderate and then moderate to severe disease in 2010. Galantamine, rivastigmine, and few others. NMDA receptor antagonists have been approved in 2003. So most of the time, the clinician, depending upon the condition, prescribes either acetylcholine esterase inhibitor or NMDA receptor antagonist or a combination of the two, right. Now, this acetylcholine esterase inhibitor is, the way it works is that it increases the level of acetylcholine in the brain, right. So, remember we talked about acetylcholine level could be reduced because of several different reasons, but scientists have not been able to find a way to increase the production of acetylcholine in the brain, right. But acetylcholine and all the neurotransmitters, once they are released in the synaptic cleft, that is the space between the two neurons, there they are degraded by different kinds of enzymes. Acetylcholine is degraded by acetylcholine esterase. It is just an enzyme that degrades it, removes it, right. So, simple idea was, okay, if you can inhibit that process using the acetylcholine esterase inhibitors, then you can prevent the degradation of acetylcholine, and thus you can increase the level of acetylcholine in the synapse, right, and hopefully it will be beneficial, and yes, that has been useful. But all these just you know just slow down the progression of the Alzheimer's disease. None of them prevent the Alzheimer's disease, right. NMDA receptor antagonist, it's a kind of particular kind of glutamate receptor, and glutamate is the primary excitatory neurotransmitter in our brain, right. So, this is one kind of glutamate receptor. Inhibition of this is helpful in case of Alzheimer's disease, right. But for the quite a few decades, people have been trying several different approaches, right. One of the approaches is the immunotherapy. Immunotherapy says that, okay, simply if you produce antibodies against amyloid beta in the brain, in the body, then those antibodies can clear the plaques and one will be free of Alzheimer's disease, hopefully, or at least it will reduce the symptoms, right. You are more familiar with the, if you don't have biology background, at least the COVID situation you are familiar with, right. People, you got the vaccination produced antibodies and other responses in the body that eventually killed the virus. So, similar logic was used here, and this particular antibody was produced, active immunotherapy studies, this particular peptide was used, and it was effective in reducing the plaques and improving the behavior in terms of memory. But a small percentage, which is actually not really too small, quite a bit, 6% of the people developed encephalitis of meninges. So, this was major side effect and it was discontinued, right. Although it was effective, but because of this it was discontinued. Eli Lilly developed this particular antibody which did not show much benefit, so it was discontinued. This antibody was produced by Biogen. It was approved in 2021, pretty recently, but then for some reason the company discontinued it because of sales not being that high. Currently, this is one approved in 2023 that is being used. So, at the most one can think of that you have acetylcholine esterase, you have NMDA receptor antagonist that blocks the NMDA receptor activity, and you have these antibodies that are used in case of Alzheimer's disease, right. But again, none of them can cure the Alzheimer's disease. Falstein test, everybody familiar with, simply MMSE, right. Mini mental state examination, MMSE, right. It measures orientation, registration, attention, calculation, recall, language and copying, and gives a score out of 30, right. It is a pretty simple test to do. It is available on the internet, anybody can do it, right. And it turns out that this is what, if somebody goes to a clinician and says, I have memory problem, I tend to forget things and this and that, and which is a routine basis, not once in a while but pretty often, and it's creating problem for me. So clinicians, attendant will do, okay, let's do MMSE test, and if that turns out to be about 24 to 30, the score, that will be considered normal. 19 to 23 MCI, mild cognitive impairment, 10 to 18 score will be moderate, and less than will be severe cognitive impairment, right. But remember, this does not tell you about particularly Alzheimer's disease, right. The scores can be slightly different, people define in different ways, but ballpark is about the same. For example, some people will say 0 to 17, one can say severe cognitive impairment. But this will definitely tell about or provide good indication of cognitive impairment in the person, right. Once that is confirmed, then clinicians use several different kinds of investigations to figure out whether it's Alzheimer's disease, whether it's vascular dementia, whether it's frontotemporal or something else, right? And then the treatment accordingly goes on. So, at NBRC, we did quite a bit of work with Alzheimer's disease using animal model system, right? And I thought I'll just show 2 slides about it. One is that this particular peptide. Now remember, this is problem with most of the neurological diseases, that a lot of drugs do not cross the blood-brain barrier, and hence even if they are very effective, they cannot reach in enough concentration in the brain and cannot provide the beneficial effect, that is the desired effect, right. So, this is a very small peptide, and peptide everybody understands, just amino acids, 1 or 2 amino acids, sorry, 2 amino acids will make one peptide, 3 amino acids will make another peptide, and things like that. So, anytime you have more than one amino acid, it is a peptide, right. So, it is a cyclic GP, which is a particular derived from insulin like growth factor, a particular protein, that we figured out that if you inject this in the mice that are designed to develop Alzheimer's disease. So, some way their gene gene thing has been changed such that they develop Alzheimer's disease, right. Basically, the mutant that we talked about, APPPS1, these are the ones that are put in the mice, and they produce a lot of amyloid beta, and the mice develops Alzheimer's disease, right. So, this develops plaques. So, all these that you can see, the whatever green light spots, these are the plaques, and we found that if you inject CGEP, not inject actually, that's wrong, it's given intranasally. So, a lot of us may be familiar with that. If something is absorbed through the nasal epithelium, that bypasses the blood-brain barrier and goes to the brain directly, right. So, this being a very small peptide, just 2 amino acid, which is really very small, can be absorbed by the nasal epithelium, and it can go to the brain, bypassing the mechanisms that are involved in preventing using blood-brain barrier, right. And we figured out that yes, it reduces, particularly in the hippocampus we looked at, it reduces the number of plaques, right, and the quantification and all that. Then the question was, okay, can it improve memory in the animals? And yes, there are different kinds of memory tasks that can be used in the animals. One particular one is uh, Morris Watermaid's task, which is used to look at a particular kind of memory called spatial memory, right. And this shows that the animals that did not get the particular compound, that is the CGP in this case, they had impaired memory, but the animals that got the CGP had very good memory, right, as good as the controls. And by several other measures, these are different measures of the same thing. To be more precise about the conclusion, one has to use several different measures of memory formation, recall in this case. So, several different measures showed that yes, CGP is effective in improving memory in the animals which are designed to develop Alzheimer's disease, right. But again, I must say that this is not the first study or only study to show that a particular compound is effective in Alzheimer's disease case. There have been several different compounds identified that have been, at least in the laboratory setup and to the using the animals, have been shown to be very effective, but they have not gone to the clinical trial and not been successful in the clinical trial. So, that basically tells us that yes, we can use the in vitro condition or the animals, but they are not same as the human body conditions, right. They cannot exactly provide the same condition. So, that is why most of the drugs fail in the clinical trial, right. And anything that we use as a medicine, as a pharmaceutical compound, has undergone a clinical trial in terms of safety and beneficial effects and side effects, only then it comes out in the market. So, MRI, particularly relevant to this session, MRI has been used in a lot of neurodegenerative conditions. Neurodegenerative condition simply means that the neurons die, degenerate, right, and that leads to the problem. It's a non-invasive, so particularly useful, right, because if there is something in the related to the peripheral body, you can take out the blood and get a lot of information what's wrong with the body. But that same thing you cannot do with the brain diseases, right, because at most you can take out CSF, which is again not very uh, not a easy procedure, but also it does not give a lot of information in every neurological condition. So, being non-invasive, MRI has been used a lot in in a lot of different conditions. Actually, there are lots of neuroimaging initiatives. One of them is Alzheimer's disease neuroimaging initiative ADNI, that is actually a collaborative effort in North America, particularly US and Canada, right. Started in 2004 and now it's in the fourth phase, which is 2022 to 27, right. So, they have collected resting state MRI data and clinical data, as well as a lot of biological fluids, for example, the blood as well as the CSF, and one can do several different kinds of analyses with those samples, right. So, people are using a lot of these data. So, this is the number of sites that are involved. It does not make uh, too much information, but lot of sites are in US, there are some in Canada, basically saying, right. Similarly, so they are looking at the default mode network of ventral, posterior, anterior, dorsal and ventral. They are kind of basically looking at the connectivity between and within the neuronal networks, right. Similar kind of connectivity that has been studied in case of hallucinatory neurodegenerative patients. There is, in some cases there is increase in the connectivity, in some cases there is decrease in the connectivity, and between regions the connectivity is increased or decreased influence under the particular condition. So, that is the kind of studies people are doing, and Professor Biswal will talk about more. There are several others initiative, Australian, European, Japanese and Chinese, right. India also has started this kind of initiative to understand the development of dementia, particular focus on Alzheimer's disease. And with this, I would like to end by one recommendation, that if hopefully you have read this paper, if not, you should read this paper, right. It is a very comprehensive review article, right, by Professor Biswal, that gives you a lot of information, actually as much as you need, about resting state MRI, what it means in terms of connectivity, how it was discovered and what the future holds, right. And with this, I would like to thank you for your attention, and we can either take the questions now or after Professor Biswal's talk, whichever way, fine. Okay, thank you.